thank you for the introduction, and I would also like to thank the organizers for the opportunity and also for the flawless organization of the conference so far. I have no conflicts of interest to declare, and my talk focuses on the complex interplay between our genes and the early environment in shaping toddler attentional competence. So this question is exciting for many reasons. Historically, there has been a great interest in the genetic basis of human attention, and amongst others, findings point to the involvement of the dopamine system. However, our genes do not operate in a vacuum, and so their effects can be modified by several aspects of the environment. As we heard this morning, during pregnancy, and in the early years of life, our brains develop at an unprecedented rate, making this period not only a crucial risk window, but also an amazing opportunity for interventions. There is increasing evidence that an adverse prenatal environment contributes to the risk of developing ADHD. And thus far, the most commonly studied prenatal risks of ADHD are maternal lifestyle factors such as smoking, alcohol consumption, substance use, and severe stress and anxiety during pregnancy, as well as low birth weight and prematurity as indicators of a suboptimal intrauterine environment. Maternal depression during pregnancy is also an interesting candidate because approximately 40% of mothers of children with ADHD have a history of major depression, making them 2 to 3 times more likely to have depression than women in the general population. Okay, and notably, and perhaps more importantly for clinicians, there are several aspects of the environment that can actually modify for the better the negative effect of prenatal adversity in genetically susceptible children. So for instance, parenting is a robust environmental predictor of developmental outcomes in children with ADHD. While positive parenting protects against comorbidity in children with ADHD, even when exposed to maternal depression, negative parenting is associated with an elevated level of ADHD symptomatology above and beyond shared genetic effects. In randomized controlled trials, actually parent trainings that promote positive parent-child interactions have been shown to be effective in ADHD, although effects might be indirect. That is, that they don't affect the core symptoms of ADHD, but they support the development of protective mechanisms such as inhibitory control. So importantly, currently the general recommendation for preschoolers showing signs of ADHD is that parent training should be the first line of treatment, and medication introduced only in case when parent training is not effective. At a very young age, it might be difficult though to tear apart like just general delays in regulatory skill developments from true ADHD. So nevertheless, research shows that in both cases, parent-child interactions might be key to prevent the development of later adverse outcomes. However, not everyone who experiences prenatal adversity will develop ADHD in the end, suggesting potential differences in children's vulnerability to the environment for better or worse. This is in line with the differential susceptibility hypothesis, in which children who are more susceptible to the environment, as indicated in the by the blue line in this figure, would perform more poorly in adverse environments, would perform average, that is similar to children without heightened susceptibility to the environment in average environments, and would really blossom in highly supportive or positive environments. Literature has highlighted the role of genetic factors in conferring such vulnerability. So here we examined whether dopamine pathway genes interact with prenatal adversity and the early rearing environment in shaping toddler attention. And we hypothesized that dopamine-based genetic liability will interact with prenatal adversity in negatively shaping toddlers' attention, but then a supportive parenting environment may buffer these negative effects. One sort of bottleneck we encountered is that, as you can see, we wanted to investigate the complex interplay between multiple genetic and environmental factors. And so far, traditional models have usually include a single gene by environment interaction at a time, or if you want to investigate multiple, then you would have to compose these one by one and add it to the model. So here we actually used a recently developed method that was developed by our lab's biostatistician, Alexia Jolicoeur-Martineau. And we basically used this method to analyze the complex and simultaneous interplay of multiple gene by environment interactions at a time. The advantage of this method that it confers greatly enhanced power over traditional methods, which usually construct gene by environment interactions one by one at a time. So our sample included 197 pregnant women and their children from the Canadian MAVEN study, who had information on 3 indices of prenatal adversity. These were prenatal stressful life events and maternal depressive symptoms, children's birth weight. They also had information on 5 dopamine pathway genes with known model of action, and 5 indices of early parenting behaviors, which were observed and meticulously coded. It's really a very time and labor intensive work at 6 months postpartum within the framework of recorded home visits. Children's attentional competence were rated by their parents at 18 and 24 months of age using developmentally and clinically well-validated rating scale. So as I mentioned, we used our alleged approach to investigate the complex interplay between 3 indices of prenatal adversity, 5 dopamine pathway genes, and 5 indices of early parenting behavior while adjusting for key socioeconomic factors as well as postnatal maternal depression. In the present study and using our present sample, we were not able to confirm the presence of a 3-way G by E1 by E2 interaction, or more specifically, the 3-way interaction was significant in the complete case analysis, but it did not generalize well to new observations, and neither did it replicate in the analysis where we imputed missing data. However, we were able to find consistent 2-way interactions that emerged in both the complete case and imputed models, as well as the variable selection and full models. These 3-way interactions were between prenatal adversity and dopamine pathway genes, and postnatal maternal parenting behavior, and prenatal adversity and postnatal maternal parenting behavior. The components that were driving these complex interactions were maternal stressful life events during pregnancy, the DAT1, BDNF, and COMB genotype, and maternal sensitivity, tactile stimulation, vocalization, and infant-related activities from mother's early parenting behavior. So we were able to confirm what previous studies have found, that multiple dopamine pathway genes affect toddler attentional competence, but our findings suggested that they did so in interaction with prenatal adversity and the early rearing environment separately. Importantly, these effects were already visible in very young children, that is 2 years and younger, and several aspects of the early maternal parenting behavior have been identified as potential targets for parent-based interventions. Based on these findings, we are very eager to conduct future research and use task-based measures of attention function as our participants have aged, in addition to parent ratings, and also to follow up who are the children that will eventually develop symptoms of inattention. we would also really like to find a data set where we can examine the effect of fathers and their parenting style on children's attention development, in addition to mothers, and also to look at other pathways that are involved in attention in the attention system, such as the norepinephrine system. Finally, we would like to extend this model to accommodate gene by gene interactions, in addition to the explored gene environment and environment by environment interactions. And currently we are working on a study where we examine ADHD as a multi-level phenomenon that is using clinical neurocognitive and neurobiological phenotypes in multiple cohorts. With that, I would like to thank the agencies that funded this study. You can see their logos at the bottom of the slide. I would also like to thank the members of the Wazana Lab. And I would really like to encourage you to check out our latest book if you would want to find out more about the effect of prenatal stress on child development. There's a great chapter in there by Dr. Booth and colleagues on the intergenerational transmission of parental early life stress, or you can also follow us on social media. Thank you very much.
Terrific. Thank you, Esther. That was a fabulous presentation. I see hands up already. So I'll ask Lonnie to help me monitor the room. I see Dr. King, it appears. You have your hand up. Would you like to ask the first question?
Yeah, great talk. And yeah, that's a great book. There's some really wonderful chapters in that book. Could you clarify for me, so in the slide that shows the interactions between maternal positive behaviors and the genetics, there we've got a low genetic, medium genetic, high genetic scores. How is the genetic score made? Was it a sum of like the vulnerability alleles? But then in your final slide where you summarize, you've got specific genes that seem to have the effect. Can you just clarify what's the genetic score and yeah, just reconcile that for me, please.
Sure. So maybe before starting, I would just also like to mention that Dr. King also has a terrific chapter in that book, just to advertise. And I'm going to try to do my best, but it's a very complicated method. And I wish, our biostatistician would be here to sort of complement on my explanation. But the way I understand it works is that these genes, so they are not simply added up, but they are kind of composing a latent feature, which is an artificial intelligence concept. And this latent feature basically is a higher order concept. So first in the main model, this higher order concept is part of the G by E interaction. But then this model is also able to tease out which subcomponent of this higher order concept was responsible to what proportion of the variance explained within the G by E.
I see. Okay, so in the slide with the figures, it's using the latent variable.
Exactly. Yeah, that's the latent feature.
Okay, and then it may be like the loadings of those. Like, so the DAT1 and the BDNF and the COMT may have the strongest loadings on that latent factor. Maybe that?
Yeah, exactly. That's what happens. So basically, the biggest effects or kind of loadings, I just like to avoid the word loading because it reminds many people of the classical factor analysis where, these components load on a latent factor, which was not really the case here, but they contributed indeed with the largest effects, and their effect was significant within this latent component or feature.
Okay, thanks for clarifying.
My pleasure.
Lonnie, I'm watching the time. We're right at time now, so I think we'll probably close down our discussion. I do see there's a couple more hands out. Please follow up with Esther either through the chat or find her email if you want to discuss further. So round of applause. Thank you for our trainees. I'm so impressed with your presentations from today, and I'll turn it back over to Lonnie to keep us on schedule.